Hi, college biology students. I have just a few more organelles I want to talk about, and I think most of these were mentioned, or some of them were mentioned in the video from yesterday, but I just want to make sure that you get that you have notes on these. So cilia and flagella only found on animal cells. These are not found in plant cells. Everything we've talked about so far, we would find in both plant and animal. Um, but these are only in animal cells. They're used mostly for movement, but there's a little exception to that with the cells found in our respiratory tract, which also have these cilia. So cilia are short appendages, and there's usually a lot of them when cilia are present. And single-celled like pond water organisms, for example, would use these for movement. They move them, and it helps propel them through the water and so forth. In in our respiratory tract, they trap particles that are not supposed to be there, like dust and other pollutants that might be in the air. And this is what causes you to, the stimulation of these causes you to cough or sneeze to try to expel those particles from your body. These are cells with flagella. Again, we also, usually if flagella are present, it's one or two or maybe three. By the way, we find these on bacteria as well. These are actually human sperm. They are the only flagellated cells found in the human body, or I would say in most animals also produce sperm. So those are, again, usually they're used for motility, but clearly not always. Um, The nucleus, obviously super important. I don't think we've talked about the nucleus yet. So the kind of the control center of the cell, very important. There are pores because remember our genetic material, that DNA is in the nucleus. It never leaves the nucleus, but it does copy itself into RNA, which then has to leave the nucleus. Nutrition has to come into the nucleus to fuel cellular processes there. So the pores are necessary for that. And also, so ribosomes are made in this central part called the nucleolus. And remember, ribosomes are attached to both the rough endoplasmic reticulum and also are free floating in the cytoplasm. So obviously they don't stay in the nucleus. They need to go out of the nucleus. They also leave through those nuclear pores. And just a couple of things then. Um, so cytoplasm is that gel-like fluid that has the organelles floating around in it. Technically, we say the cytosol. Actually, I should add that on here. The cytosol is just the fluid itself. I don't know that I have that on that, so I'm just going to add this for you. Cytosol is the gel. This should be hyphenated. Gel-like fluid only. And then centrioles. So these centrioles become very important when cells are dividing during that mitosis and meiosis schedule or process, I should say. All right, parts of the eukaryotic cell is what we've been talking about so far. And almost all of the things we've talked about so far we would find in plant and animal cells. The exception would be those used for motility. Plant cells don't need cilia or flagella because they don't move. But plants do have some additional structures. Animal cells also have vacuoles. They just tend to be very small. But in plant cells, they're very large. And of course, plant cells have a cell wall in addition to the cell membrane, not instead of, and then plastids. So the cell wall is this very rigid structure, and plants need that for support because they don't have a skeleton like animals do. So they have to have this very rigid cell wall. When the cells are still growing, they only have a primary cell wall because it's still forming. It can't be super rigid because it's still growing and so forth. And then when the cell is mature, it matures into this secondary wall, which is that that very rigid structure that even when the plant cell dies, we usually can see residual remains of that that cell wall. Vacuoles, we said, in plant cells are very large. We also say that when this vacuole is full of water, so it stores water and also enzymes and some waste, but when it's full of water, the vacuole itself pushes against that cell membrane. Sorry, I'm writing with a mouse because I don't have my board. So a full cell, we say, is turgid, and that vacuole is pushing against, I hope you can read this, is pushing the cell membrane actually against the cell wall. Obviously, if it's not turgid and not full, the effects to the plant would be wilting, right? So the plant would be wilted and eventually it would die. Plant cells also have things called chloroplasts. Not all cells in plants have chloroplasts. Some of them only have leucoplasts. But chloroplasts are these magical things with chlorophyll in. They're the only chlorophyll can capture energy from sunlight, so super important to life on this planet. So responsible for capturing energy from the sun. I'm just going to go kind of quickly because you should have learned this in 10th grade. Storage plastids, then called leucoplasts, are responsible for storage of starch, lipids, or proteins, usually found in the roots. They're not photosynthetic. Things growing below the ground don't need to be. So we looked at, we tested a potato for starch, found that potatoes, which grow below ground, have lots of starch. The onion cells that we tested had some starch, but also some sugars in there. But we don't see any green typically in things growing below ground. The green is characteristic of chloroplasts. Things that grow below ground don't see the sun. That would be just pointless, right? So, quick overview. I'm not going to go through all of this. Please do so at your leisure. I really like this concept map. I think it just really gives the job of all of these different organelles. I think this is incredibly helpful. So for energy, chloroplasts and mitochondria, the recycling, of course, those lysosomes, and so on and so forth, manufacturing proteins, and then lots of things involved in different areas of transport. Really quick overview of prokaryotic versus eukaryotic. I think we may take some bacterial swabs. Actually, that'd be a great thing to do either this week or next week. Sorry, thinking out loud. I have some Petri dishes. We could easily do that. And then we could look at some prokaryotic cells. I used to teach microbiology, loved it. Just don't have room in my schedule. So maybe we'll incorporate just a little bit of that into your curriculum. But prokaryotic cells, always bacterial. They don't have a nucleus. Prokaryotic literally means pre-nuclear or pre-nucleus. You then, as well as plants and fungi and all of those living things, are made of eukaryotic cells. These might be multicellular, might be single cellular. Prokaryotic are always single celled. All prokaryotic cells are bacteria, and all bacteria are prokaryotic cells. They're much smaller, much simpler. Multicellular organization. So cells, we know, are the simplest unit of that can be considered living. Cells make up tissues. We have 4 main different types of tissues in our bodies, or in living things, I should say, in general. We have connective tissue, like our ligaments and tendons, epithelial tissue, which is our skin and coverings of internal organisms. We also have muscle tissue and nervous tissue. And those are the 4 main categories. There are different types of muscle tissue, obviously, and so on. But tissues make up organs, so we like our heart and our lungs and so forth. Groups of organs make up organ systems, like the respiratory system or digestive system. And then organisms is the most complex, and it is all of the organ systems working together. And if you remember from 10th grade biology, I know we kind of mentioned this same thing. There is something called colonial organisms, and this Volvox is a really good example of one, where these are single-celled organisms that cluster together in groups, and when they stay together in a group, it forms this membrane around the colony. And once they're in a colony, the cells begin to specialize. Some of them are responsible for movement of the colony. Some of them are specialized for reproduction of the colony and so forth. So they become this, specialized within this colony. But if this envelope that surrounds them, this membrane, breaks open, they can all function individually. So scientists believe that perhaps this, colonial organism was kind of the precursor to multicellular organisms. We do believe the first organisms on this earth were bacteria. Okay, so something that you haven't talked about before in like 10th grade biology are how cells communicate with each other. So there's cell-to-cell interactions, there's junctions between cells that have really important jobs, and we're going to talk about those next. So first of all, we said that the cell membrane has it's this phospholipid bilayer, and they're held together by hydrophobic and hydrophilic properties. Get rid of this. The surface proteins of the cell actually give the cell its identity. So on your immune cells, this is a B cell, and there's specific markers like a CD20 marker, a CD19 marker, and they have different shapes and so on. So there's surface proteins that actually identify the types of cells. We know this in blood types. So there's A, B, and O, and AB blood types. So it's actually some types of lipids that are tissue-specific cell markers that give cells their identities and make them different from each other. Major histone compatibility proteins also allow the cell to recognize as self or non-self. So this is really important because if other cells don't recognize this cell as part of yourself, it will begin to attack your own cells. And that's what some autoimmune diseases are. So there's an inability of the cells to recognize markers on the cells as part of the same organism. This also becomes an issue like if people get transplants. So if you get a liver transplant, for example, from someone else, let's say you had a liver disease and your liver is not functioning properly, and basically you can't live without your liver. So you get on a transplant list, and even if it's a close relative, your body knows that that is not yourself, and it will attack and basically kind of eat away at that transplanted organ that you received. So people who get organ donations from others usually have to take anti-rejection drugs for the rest of their life. And obviously there's a lot of side effects with anti-rejection drugs, but if you have no other choice but to, if your liver's not functioning and you need one, and we can do heart transplants and heart-lung transplants, like kidney transplants, like we do a lot of really cool stuff. And so, yeah, that we have to tell the body, please don't recognize this as not self, because it will literally attack that foreign organ. So most of the time, this is really important and life-saving. But again, people with autoimmune disease, there's something not working here. It's not recognizing self as self. It's actually targeting some of its own body as non-self, and obviously that's a problem. So that's a basic introduction to the next section. We'll pick up with this later.